Hi everyone. I do realize I'm the sixth speaker of today, and it's right before the break, so I'm sorry. I already see some people getting really comfortable in their chair, but bear with me, give me some last energy for the last 10 minutes, and then we go all together to the break. All right, let's go. So today I will be presenting you plant-based scaffolds that induce serum-free cell adhesion for cultured meat. I have some spoiler alert. I'm sorry, guys. The plant-based, I cannot disclose the type of protein yet. I asked my professors. They said no. I still have to write my article, my publication, and then it's ready for everyone to know, but unfortunately not today. On the other hand, serum-free, I think during this conference, you were all talking about serum-free alternatives maybe already, but in my presentation, actually serum-free is just that I excluded FBS fully. We did not create an alternative or anything. I leave that up to you guys, but I just did not use it. Okay, it's important for the future results. So, how did we make this scaffold? See, I hope the colors there are better. Okay, don't watch this, those are better. Um, so how did we make the scaffolds? We first prepared a solution of the protein. We added glycerol and water. We heated it up, and then we put it between two parallel plates. And in the middle, there is like a spacer of 1.5 millimeter. So all the scaffolds later on have exactly the same thickness. Then there was a cross-linking step, and eventually we like punched out all these circles with the same diameter, because actually we want to use them in a well plate. Also, what is important to know, this is a non-tissue culture plate, because I really want to only see the cells on top of my scaffold and not on the bottom of the well plate. So I did an extra sterilization. That's why my question was for the previous speaker. Although we tried to work as sterile as possible in the first steps, we see it's not sufficient. So we added a sterilization step with ethanol for 24 hours. And before we use it, we wash it away with PBS. Okay, so now we have the scaffolds in our well plate, which are hopefully very sterile. So we can move on to the cell seeding. So like everyone else, we've been using C2C12 cells. We did a drop seeding, which means we add like a very concentrated form of the cells first. We incubate it for like half an hour, and then we add the rest of the culture medium. And we incubated it for 72 hours. What was my setup in these tests? First of all, of course, the scaffold that I just showed you, but I also wanted to have like a reference to see, is my setup working? So we just use gelatin, the GelMa, the methacrylated gelatin. It's not our goal to use it, we just use it as a reference, right? And then, of course, the non-tissue culture plate as a control. Next, we checked for cell attachment. My scaffolds are not transparent, so you can not see the cells just under a microscope. So what we do is we perform a cell titer glow assay. I don't know if you guys know it, but it's amazing because it's a really easy assay. This is how it works. Actually, it reacts with the ATP that is present in your cells, and it generates a luminescent signal. You just add it and you measure it. What do we see in these graphs? Just some short explanation. CE stands for cells in serum-free medium, so just FBS excluded. Then we have CM, that stands for cells that were used with serum. Of course, we use serum as well as a comparison, right? And then you have three colors. In green, we have just the culture plate with serum, without serum. In orange-ish, you have the gelatin. So the gelatin with serum, without serum. And then we have the protein, also with serum and without serum. Okay? So what do we clearly see? Without serum, so the CEs on the plastic and on the GelMa, there is like 0 signal, right? If we add serum, we do get a nice signal. Also, surprisingly, the non-tissue cultured plates, if there is serum, yeah, they still attach. But what was the best result, actually, is that on our protein, they were able to attach to the scaffold, both with serum and without serum, which was not the case on the plastic. Okay, not surprising, but it's also not the case in the gelatin, which was actually quite surprising. So, to confirm this, we also did a visualization. On the left, you see the gelatin scaffold in serum, and then you have the protein scaffold in serum, and then you have the protein scaffold in serum-free. And in all 3 cases, we see indeed cells attached to the surface of the scaffolds. Sorry. Another thing that was very remarkable, and that we actually saw only after some months of testing, is that we decided to also check the bottom of the plate where the protein scaffold was present. And what did we see? There were also some cells attaching to the bottom of it, which made us think something might be leaching from the protein scaffold that can induce cell attachment in a serum-free environment and a non-tissue cultured plate. So we wanted to confirm this. So what did we do? We took our scaffolds, like we always prepare it, and we just put it in serum-free medium. No cells there. We just let it soak for some days. And then we took that medium without the scaffolds. We just took the medium and we used that one for cell seeding, right? And again, this is serum-free medium, but now I call it like enriched medium because we have the possible leachets from the protein. So what did we see? Indeed, if we use this enriched medium, we have the protein leach cells in serum-free. They give almost an equal signal as when we use serum. Of course, this is after 24 hours. It's not possible on a long term yet, but this really shows that there is something there. So our conclusion there is, okay, apparently cell adhesive components leach from the scaffold, and now we are identifying or trying to identify these components or these components through proteomics. The second part of my presentation, still about the same scaffold, because of course now we really like it. But now we move on to the microcarrier story. We all know, okay, we want to move from 2D to 3D, and we decided to create these plant-based microcarriers. Why did we want to do this? Because they are edible, fully plant-based, so you don't need any cell detachment anymore, right? You can just incorporate everything in your end product. This is a picture of these microparticles, and on the left side, you see how they look like in suspension. And after some minutes, the particles, they already sediment, which is a very good property because actually we want to be able to remove and refresh the medium once in a while. So if the particles sediment, we can change the upper part of the medium. Okay. So we tested this as well, and we compared it with the Cytodex microcarriers as again as a reference. And then on the other hand, our plant protein. So we used like a spinner flask, and this work was done together with Maria Olenek. I think some of you guys will also know her. So we put all this in the spinner flasks. I have to mention here, we used bovine myoblast cells here, which is more relevant than the C2, C12s. But in here, we still used FBS because the myoblasts are way more sensitive, and we really need first like a proper serum-free alternative to really move on to the serum-free condition. But in here, the purpose was really to prove the concept of the plant-based microcarriers, right? So what do? We seeded 1 million bovine myoblast cells, and then we kept them in culture. As you can see, actually really promising results, right? We will not deny that the yield is still a lot lower, but on the other hand, these were the first tests, so we are still optimizing all the parameters from a spinner flask, the sizes of the microcarriers and the shapes. So it's an ongoing project. But I already wanted to share these results with you guys. All right. Congratulations. You all sat through this presentation. Most of you are still awake. It leaves me to thank my team. This is really a teamwork. And actually, I'm part of 3 different labs. It's a food structure lab, a cellular lab, and a polymer lab. I have some pictures of the nice groups there. And in general, I am part of the custom meat project, which is a big project together with Ghent University and KU Leuven, and these people, their names. Thank you.
Thank you very much. Are there any questions? Yeah?
Oh, I can just use this one. Sure. Thank you for the excellent presentation. If I just had a suggestion for you, because I've had similar problems as you on non-treated wells. So actually there is an ultra low adhesion wells there where they coat the wells with a coating so that nothing sticks to the bottom of the wells.
Yeah.
So if you would really want to seed your cells on well plates that are non-treated, ultra low adhesion, then you can use those.
Yeah, actually, we also used those, but then we started like rethinking, like, okay, but if it's already serum-free, actually the effect that we see on the bottom is also interesting.
Yeah, for sure.
But thank you for your suggestion.
And then, is there anything more that you can say about your plant protein, other than what you've already said?
No. I'm sorry. I really hate to do this. I always say that it's so sad for science that we do this. But okay.
Thank you for your talk. Have you looked into potential cytotoxic effects from glycerol? When I read the literature, I hear that it has some, and then when we put it in our plant protein scaffold to make it less brittle, it seems to have a little bit of a cytotoxic effect.
Yeah, we tested several concentrations of the glycerol, and then we chose a concentration where we didn't really see a difference anymore. Yeah, but for sure you cannot go too high because it's indeed toxic.
Thanks for the great talk. Did you also try enriching the medium with just the PP, so the plant protein extract, instead of the scaffold you made?
Yeah, I'm not sure if I'm allowed to open that can of knowledge, but yes, we did, but it doesn't work.
I see. Okay, thanks.
Yeah, thanks for the talk. I may have missed that part, but how can you be sure that you are, because you observe adhesion, but it takes quite a long time, that it's not coming from extracellular matrices produced by the cells?
So you were one of the guys sleeping right in the middle? No, I'm joking, I'm joking.
I may have missed, I mean.
No, I'm joking. That's a good question, but that's why we have the gelatin scaffold as a reference, where the cells don't attach to. And they are in the same environment, so why would they not produce their ISCM on the gelatin, but they do it?
Yes, and I think we have to discuss, because you can imagine a modulation by something that is in the well, modulating the expression of a new protein or something that can help adhesion.
Yeah, indeed. Do you think it's possible that we would know it through the proteomics? I think so, maybe. We will see. I don't know. Thank you.
Okay, well, thanks again, Indi.